Okay, so I will start with an introduction about the topic, then Daniele will talk about the computational and technical part, and I will then come back in the end for the clinical applications. So the project, as mentioned, is focused on the development of new technologies to try to exploit the power of single-cell RNA sequencing in clinical nephrology. And I would like to start with the very basics. This is the central dogma of molecular biology. And we know very well that in basically any biological process, the biological information needs to be transferred from the genome to RNA and then to proteins, which then will lead to the function and determine the phenotype in different biological systems. So today we will focus on the transcriptome, basically on RNA, and there are some good reasons to do that. The first is that current available technologies are particularly powerful in analyzing nucleic acid-based information. In this way, it's much easier to get robust, comprehensive, and quantitative data. But, obviously RNA is much more dynamic and regulated compared to DNA, and therefore this is probably a more meaningful information for the majority of the disease. I will not go into the detail of the different ways of measuring or determining the transcriptome, just a brief overview about the technology we are using, RNA sequencing. You basically start with a sample from a patient or a clinical condition. And the very important first step is obviously to isolate the RNA. And to do that, you basically have to destroy the tissue sample and to isolate the RNA you're interested in. Then you go to the next step where you generate a cDNA, a copy cDNA, which is then sequenced with various technology, and then putting together these fragments of sequenced information, you can map this to the genome and basically have an idea of the transcriptome in the whole sample. This is a very powerful technology, and it can be used in many different ways. For example, in this study, we performed transcriptional analysis by RNAseq in a cohort of patients after kidney transplantation, and combining information obtained in different biopsies at different time points. And by integrating this information, we were able to generate a molecular map of the transcriptional trajectories, determining the fate of this kidney from the very early response to ischemia reperfusion to patient recovering completely and having a good long-term kidney function, and other patient progressing toward a chronic injury and therefore to deterioration of the kidney transplant graft. So this is obviously a powerful technology, but on the other side, you notice that it has a lot of limitations. So as I mentioned before, the first step in this experiment is to destroying your tissue to extract the RNA. So if we look at the beautiful kidney with many different cell types organized in a very precise way. So the first thing we do is actually to destroy this. So we lose a lot of important information. And for this reason, new technologies were developed, which are more focused on single cell analysis. So instead of destroying the kidney or the tissue, you basically dissociate it to get a suspension of single cells. And then by applying fluidics technologies, you basically encapsulate these individual cells with beads, and you can perform your sequencing analysis within this vesicle. And so you can get the transcriptome of the individual cell type. So instead of having a milkshake with very general information, you can have here the information you obtain from the individual cell types. So this dramatically increase the granularity of your research, and is changing many fields of biological research. And just to give you an example, I had the chance to work on this technology when it first was applied to the kidney when I was in the US doing a postdoc. And there we basically did the first experiment where we applied this technology to just a normal mouse kidney. And by doing this, we identified that there are more than 30 different cell types in a normal kidney. We found that there are some differences between male and female in very specific segment of the tubule, which were not known, or we characterize, for example, this very critical part of the kidney where the conjunction between the distal tubule and the collecting duct, where the cell population are particularly challenging to distinguish. So obviously the things become even more interesting when we move to kidney disease, particularly in a translational or clinical perspective, because we know that actually more or less all kidney disease are associated with very particular cell types or cell states. So the cells get damaged or they enter into a particular cell state. Here as an example, if you have a tubular injury, this is a normal epithelium. After tubular injury, you have this damaged or dedifferentiated cells. And we know that these cells are not only important because they lose their normal properties and function, this contribute to the dysfunction of the organ, but there is some evidence that they also contribute to the inflammatory response leading to chronic inflammation and fibrosis, which definitely lead to kidney failure. So when we apply this technology to models of acute kidney injury in mice, we were able to, by single cell analysis, to really characterize all these different cell types. And particularly interesting was this population of late injured proximal tubule cells, which were labeled by or marked by VCAM, which turned out to be an interesting gene in this particular setting. And you know, kind of bring the first evidence about these dynamic changes in the response to acute kidney injury. So obviously we would like to use this in patients, but there are some limitations to that. The first is that the processing of the tissue, do you need to do that, is quite difficult to apply in a clinical context. And on the other side, you generate a lot of data which are not so easy to process and to analyze. So the general idea is that the technology we have nowadays for single cell is not directly applicable in a clinical setting in the everyday clinical business. So one approach that could be used to make use of this information is to take the information you get from a limited number of patients you can analyze with this technology, and then based on this prior knowledge, develop some computational tools based on statistical modeling or more advanced artificial intelligence-based tools, and to use this information in the context of you know, patients or patient cohorts. Also, in this case, there are some challenges. So first of all, you have to have an atlas, a reference atlas, to map these different cell types, particularly these damaged cell types. It's not something that you can find in the normal tissue, so it's difficult to get this information. And obviously we are dealing with different levels of complexity. So, you know, we know from clinical experiment that it's already difficult to identify groups of patients. Now we are adding here maybe tissues or samples at different time points. And for each of these sample, you get thousands of cells. And from each cell, you get maybe the information about 20,000 genes. And so how to integrate this information is obviously not so easy. And you also have to consider that in the end, in the clinical context, you have to deal with this huge information, but then to tailor it down to a situation where you usually have a very limited number of patients in a cohort in many clinical conditions. And ideally in the future, maybe to the single patient, if you want to really have a personalized medicine approach. So these were the questions we had. And with this question, we went to the colleagues at the IDSIA and we asked for solutions. So give the words to Daniele for the next steps.
Good morning, everyone, and thank you very much for the nice introduction. So as announced, I will go a little bit more technical. I will try not too much. And my starting point will be bulk RNA sequencing. So the typical outcome of a study conducted on some biopsies, doing bulk RNA sequencing on these biopsies, is a data set which for every sample that we can see here on this rectangle contains a description in terms of genes. And typically for each single sample, and you might have some tens or hundreds of samples, you will have a description featuring more or less some 60,000 genes. And this is highlighted in this picture. As you can see, this data set is highly rectangular, and working with these kind of things is usually complicated. So it is desirable, if possible, to try and have a more compact representation of this data set. Visually, to make this data set closer to a square, like in this case, where we will have the samples but every sample will be described not by 60,000 genes anymore, but by a limited number of variables. And the PLIER algorithm is an algorithm which allows to do this operation. It was published in 2019 in Nature Methods, and PLIER algorithm is also the basis of it's what we use in our work. So essentially, with PLIER algorithm, you can learn a transformation which allows to perform this dimensionality reduction, going from a high dimensional dataset, this highly rectangular dataset, to a smaller dimensional one, this B dataset. Technically speaking, what the algorithm does is a matrix factorization. These datasets are basically matrices, so we can decompose a dataset into a product of 3 different matrices. So let's imagine that we have already trained our algorithm, and we decompose a new dataset into this product, C, U, and B, where C is a matrix which contains biological information. And this is something that we also provide to the algorithm for the training. The second matrix is actually something that we learn during the training, and which contains information about relationships between new variables and the biological information. The biological information is in terms of pathways. So basically a pathway is what describes some kind of biological process. And what we fit into these matrices is a list of genes which are involved in these pathways. And as a last element of the product, we get what we will use as a final outcome of the transformation, which is this lower dimensional dataset, where now our samples are not described anymore in terms of genes, in terms of a lot of genes, but in terms of a limited number of variables that we call latent variables. And this typically is in the number of less than 1,000 variables. The most interesting thing of this procedure is that between Y and B, there is no loss of information, which is key for what we will do in the following. So let's now explain a little bit how we got from the PLIER algorithm, which is the method that we use, from K-CLIER. K-CLIER is a transformation that will apply to clinical studies in the second part of the presentation. So we feed this PLIER algorithm with 2 elements. The first one being a large data set. This is in the training procedure. And the second one being a list of single cell signatures describing biological information. Let's focus on these 2 elements. The large data set that we use for the training is a data set that we can find on the Cancer Genome Atlas, TCGA, which is a project aiming at putting together a lot of data, which are open access also, and describing patients affected by cancers. And in this data set, we can find more or less 11,000 sequencing, bulk RNA sequencing for patients affected by 33 different types of cancers. It is worth mentioning as well that the dataset also contains biopsies from healthy samples. As a second element, we provide to the algorithm some biological knowledge. And here there is a novelty with respect to what PLIER was conceived for, because the P of PLIER stands for pathway, whereas in our case, K-CLIER has a C, and this C means cell. The information, the biological information that we are providing to the algorithm is some single cell information. So it's pathways which are specific from certain type of cells or cell states. And these single cell signatures is something that we extract from studies done at the level of single cell RNA sequencing. So we extracted 725 single cell signatures from recent publications. And these ones are associated in most cases, were retrieved in most cases from disease biopsies. Here, the height of these bars is proportional to the number of signatures that they contain, where disease biopsies means either cancer or chronic kidney disease or even diabetic nephropathy. But we also had some biopsies coming from normal samples, and it is worth mentioning that we also had some signatures describing some urine samples. In terms of cells, we have a high presence of lymphoid and myeloid cells, but also proximal tubules, tumor cells, and a lot of groups more that we will use also in the rest of the presentation. Something very important is that these signatures are tailored for studying kidney. So all of them are single cell signatures describing some kidney biological process. Also, they all come from human samples. It is also worth mentioning. Now, with these elements, we can train the PLIER algorithm, and the output will be our K-CLIER, where K, of course, stands for kidney in this case. And with this algorithm, with this K-CLIER ready, we can apply K-CLIER to a new dataset, a new high-dimensional dataset, from which we get, thanks to K-CLIER, a lower dimensional representation. And the output of this will be, as we said, a dataset containing these latent variables. In our case, it will be 700 latent variables, which reduce from more or less 60,000 genes. Well, this can be, of course, the input for many things: machine learning, data analysis, clinical interpretation. But before moving to that, I would like to spend some time on one of the key features of this method, which is interpretability. A part of these latent variables is interpretable in the sense that it's easy to see a link, a direct link between the latent variable itself and biological information. And on the right of the slide, we can see some examples of these. For example, our LV406, of course, LVs go from 1 to 700, is associated to a signature of cell type stroma. Another one, a little bit more complicated, is associated to 4 different signatures, all belonging to lymphoid cells. Once again, the height of these bars is proportional to the importance in this case, or the association in this case, between the latent variable and the given signature. Also, we can have a case like this one, like LV025, where we have a lot of signatures which belong to several different groups. And in this case, in particular, two groups, like proximal tubules and tumor. This coexistence between these cell types inside the same latent variable is not trivial, actually. It has a meaning. And we will see this LV025 again in the rest of the presentation, and especially in the second part. Now, with this in mind, with this in mind, we can perform a validation of this method, of course, because as every method before being used, it needs to be validated. And we perform several different validations, some based also on machine learning models. And here, for time reasons, of course, today we will only present one of these. And this is one that we performed on TCGA itself. This is something that we can do since our dimensionality reduction method is an unsupervised method. So as it often happens in bioinformatics, we can do that. And in particular, we decided to focus on a subset of TCGA. And the subset was, of course, kidney samples in TCGA, since our interest is working with kidney. So this subset of TCGA contains 4 different types of cells, 3 belonging to 3 different types of cancers, which are very different, profoundly different, as we can see also in the images on the right. And these cancers are clear cell carcinoma, papillary carcinoma, and chromophobe carcinoma. And besides this, we also have some normal samples. Now, we applied our K-CLIER transformation to the initial high-dimensional data set containing these samples. And what we obtained was our 700-dimensional description of this data set. And here we visualize with a method called UMAP, which perform an additional dimensionality reduction to 2 dimensions. We see an image in 2 dimensions of our data set obtained after the processing with K-CLIER. And what we can see in this image is some very, very clear structures that we identified as clusters by using some clustering machine learning methods. So we have 4 clusters. And one reasonable hypothesis would be to hypothesize that each one of these big structures corresponds to one histological type. And we see how we can validate now this hypothesis. So what we tried to do was try and characterize each one of these clusters in terms of latent variables. So for example, we can focus on one of the clusters and look at what latent variables are upregulated or more expressed, if we want to use a gene language, within the cluster than outside. And in this way, we can produce this kind of plots where we can see, for example, that if we focus on cluster 1, LV599 is the most significant latent variable for characterizing this cluster with respect to the others. And this is associated to both tumor cells and proximal tubule cells. Statistically speaking, what we are doing is doing multiple tests for trying and finding the most significant latent variables in characterizing each cluster. Now, if we have a closer look to, for example, the first cluster and LV599, and we look inside the structure of association of LV599, what we find in this case is many signatures which are associated to clear cell carcinoma. So we could conclude that actually this should be the cluster of clear cell carcinoma samples. And this is actually very interesting because to get back to one of the images that we had in the introduction, we are using bulk RNA sequencing. So here we are using a fruit juice, and what we are seeing is an image of real fruit from this kind of plots. If we do the same operation on the second cluster, cluster number 2, we see something similar. So we see that in this case, the latent variable, which let's say characterizes the most the samples inside the cluster, is associated to papillary cell carcinoma. So these should be the clusters of patients affected by papillary cell carcinoma. And if we do the same operation for the other two, with similar consideration, we could say that this one is the cluster for chromophobe cancer, and this one is the cluster for normal samples. And there is a very simple way of validating whether our hypothesis is true, which is just coloring these samples, since each point is a sample here, using its true histological type label. And what we can see in the bottom part of the slide is precisely what we expected. So for example, this structure is highly uniform in histological type, and it's also associated to clear cell carcinoma, as we inferred by starting only from bulk RNA sequencing data. This carries two important messages. And the first one is that our interpretation is consistent with true histological type. And the second one is that the information about histological type, which was contained in the initial high-dimensional gene dataset, was preserved while applying our K-CLIER dimensionality reduction method. With this validation, as I mentioned, we performed several others. But instead of going into details of the other validations, I would like to conclude with one slide on this technical part, and then give back the word to Dr. Cippà for the applications of the method. So the main messages from this part are that our K-CLIER is a dimensionality reduction transformation. It was learned on a large dataset on TCGA and can be applied to any gene expression dataset that we have. The most interesting thing is that there is no loss of information in doing this operation. Also, a part of the data are directly interpretable in terms of cell type or cell state. Also, this is by construction tailored on kidney disease, since we fed the algorithm with kidney-specific information. And the last point, this can be applied also to very small dataset, which is a typical clinical use of this kind of methods, and which is also the topic of the next part of the presentation. Thank you.
So it seemed that they solved most of the questions we had. So we went back to our main focus of research, which is again the acute kidney injury and the transition to chronic injury or chronic kidney disease. And this is the model we have today. This is a normal kidney with the tubules and the vascular structures. After acute kidney injury, some of these tubular cells would die. Some others will respond with what we call a repair response, and will start dedifferentiate, proliferate, and restore the tubule. So if everything is going well, we'll go back to a normal kidney. But we know that in many clinical settings, this is actually not going on in a perfect way. And some of these cells will remain in this undifferentiated state and start to be dysfunctional and produce factors that will stimulate fibrosis and chronic inflammation. So having this new tool, we wanted to see if this model, which has been basically designed based on experimental data in animals primarily, could be applied to identify the different cell types in human sample and to validate the model. So first we focus on chronic kidney disease, kind of a static picture, because it's a kind of an easier setting, and we decided to use diabetic kidney disease as a model. We had access to a cohort of 37 patients with biopsies with diabetic nephropathy or controls. Please note that this is a very small number of patients compared to the numbers we heard this morning. And we had RNA sequencing, bulk RNA sequencing from all these biopsies, and obviously the histological evaluation. We applied a K-CLIER, and we correlated which latent variables are associated with fibrosis or no fibrosis. And as you can see here, many latent variables were associated with, for example, with fibrosis. And you can analyze this in detail to see which cell types are associated with the latent variables. We'll just pick up a few examples to give you an idea. These 3 latent variables. So latent variable 182 was a latent variable strongly associated with lymphatic cells, and particularly if you look exactly, these are all B cells or plasma cells. And this was actually very much consistent to our previous publication, where we described that these B lymphocytes are important component in this transition from acute to chronic kidney injury. Another cell type or latent variable which show up was LV627, which is strongly associated with stroma cells. And if we look more in detail, you see some population of fibroblasts or myofibroblasts that we are recently described as strongly associated with a chronic kidney disease in this Nature paper. Other latent variables were a little bit more complex. For example, this LV21 associated with different cell types. And if you look a little bit closer, we see the association with immune cells, with some epithelial cells, and also with endothelial cells and myofibroblasts again, suggesting that one single latent variable here could actually reflect the combination of different cell types, but we know are strongly interacting, building these cell communities, which are probably important in these biological processes. So this was not particularly surprising that we would find these cell types here, and I think it was more the proof that we can apply this technology or this tool also in the experimental set or the clinical setting we are interested in. What we are more interested in is to have a kind of predictive tool. So the question is, can we identify cell types occurring in the early phase after injury, which are predictive to their transition to chronic kidney disease? And this could also be important to have a prognostic tool, but also because this might be a good population to be targeted in a therapeutical perspective. So to do that, we use a cohort of kidney transplant patients where we have repetitive biopsies in the same kidney basically over time, and particularly 3 and 12 months after transplantation. So what we could do here is to analyze the biopsy at 3 months and look at the latent variables there, and to correlate this with the presence or not of fibrosis at 12 months defined by conventional histology. So we could analyze this more in detail, but I think we have to focus on some aspect. The strongest association with fibrosis was with this latent variable related to stroma cells, and particularly this fibroblast 4A identified by Kuppe. This is not surprising because obviously if you have fibrosis at 3 months, this is an irreversible process. It's very likely that you will have it later on as well. But then we also saw that in the patient not having fibrosis, we have this association with different cell types, including, is this working? No. It's not working. Okay. Including, importantly, some proximal tubule cells, which, if you look more closely, these are basically normal proximal tubule cells we obtain from normal kidneys. But there are also some latent variables, the LV025, associated with proximal tubule cells, but are associated with fibrosis. So we went further on that and to really characterize it. And if you see, at this time point, we find here that some proximal tubule cells identified here are exactly the VCAM1 positive cells that we have already characterized in the mouse model, as I presented in the first part of my presentation. And we see also several proximal tubule cells, which have been identified in this data set by Legui and al. This data set of Legui is actually one data set we generated very recently. because we wanted to have a good single cell map of patient having acute kidney injury in the very early phase after acute kidney injury. This is a clinical setting where we usually do not perform clinical biopsies. And we have the chance to do some biopsy in 5 patients with severe COVID-19 in Geneva during the first waves of the COVID pandemics. And this generated a very interesting data set in patients in the early stage of the acute kidney injury, with a particularly important information related to these damaged proximal tubule cells, which we characterized as being dysfunctional in many ways. They reduce the possibility to do gluconeogenesis, their metabolic function was altered, and they had an increase in this extracellular matrix score, which is expected to signalize cells triggering fibrosis as validated by conventional or by immunohistochemistry. So basically, by combining new data set from single cell in a limited number of patients and then applying the K-CLIER in a larger cohort, but still manageable numbers in a clinical setting, we confirmed that the model we developed in the mouse models based on the central role of proximal tubule cells being damaged in the early phase, but then if persistent after repair, being the key player in producing cytokines and other factor producing inflammation and fibrosis, can be confirmed in human. And I think this is the first time that this is really kind of validated in a clinically relevant setting. So the last point then for this presentation was the fact that we wanted to, in a clinical perspective, we would like to use this technology not only on bulk RNA-seq from biopsies, which is still, a limitation to the broad application. And it would be better to be able to have a similar technology based on urine or blood samples. And we decided to test the K-CLIER also in this setting by using urinary bulk RNA-seq. And we did it in a cohort of kidney transplant recipient, 57 patients in this case, where we had the bulk RNA-seq and a correlation with the histological diagnosis at the same time point. And in this case, we were interested to see if we can find a sign of acute rejection. So I will not go into the detail, but we looked at latent variables associated with rejection. And as you can see here, we have here this strong association with latent variable related to lymphoid and myeloid cells. And if we go more in detail, this patient had a signature compatible with the presence of CD8 T cells or M1 macrophages, which are known to be key players in acute rejection. So it seems that the same technology can be applied also on clinically relevant samples. So to sum up, I hope that we convinced you that we have a computational tool to link the granularity of single cell information to clinical outcomes. We can reduce dimensionality without losing important information, and we can interpret this data. I think this is a major point of this tool. And with this, we can identify different cell types, cell states, communities by using bulk RNA-seq. The approach is generally applicable. What we did is to tailor it for the kidney, performing several validation and applying this to kidney biopsies and urine samples. If we think in terms of application, obviously one option would be to have a diagnostic or prognostic tool, ideally in blood or urine. But in my personal opinion, the real strengths of this technology or this tool is more related to translational research. I showed you that we could validate experimental findings in patients, something which is sometimes very difficult to do. For example, here, the role of these altered proximal tubule cells in chronic kidney disease progression. But obviously, the other way around, we could generate hypotheses for further experiment. And I think that this is a critical point because these single cell technologies and other technologies are really progressing extremely fast in basic science, but the challenge now, and I think it's very good that today we are focusing this kind of problems, is how to translate this into clinical application. And we know that this doesn't occur just by itself, but it needs investment, research, but also good ideas. So thank you very much.